Okay, so welcome to this next video in which we are discussing lipid modification of polypeptides. Okay, right. So we are now in the process of discussing palmitoylation of proteins, which is the adding on of palmitic acid molecules onto proteins. Okay, right. So there are two ways that you can add palmitoyl groups onto proteins. Okay, they're always added onto cysteine amino acids. So let me remind you of the structure of a cysteine amino acid. Okay, so let's start with what is known as S-palmitoylation. Okay, and we'll see in a moment that there's also N-palmitoylation, but we'll start off with S-palmitoylation. Okay, so this occurs onto cysteine amino acid residues, which are in the middle of the polypeptide. So let me make this crystal clear. Okay, so it would occur, for instance, to an amino acid that was right in the middle, like so. Okay, not at the amino terminus. If you have a cysteine amino acid as position 1 amino acid, then what's going to happen is you're going to get N-palmitoylation, okay, which is very similar to N-myristorylation. But if the cysteine amino acid is within the polypeptide, i.e. its amino terminus is not free, then you're going to get S-palmitoylation. So let's draw the structure of a cysteine amino acid that is deep within the polypeptide. Okay, so this means that its amino group will be involved in binding to the carboxylic acid group of the amino acid in front of it. Okay, then here's its alpha carbon, here's the carboxylic acid group, and its carboxylic acid group will then be linked to the amino group of the next amino acid along. And the R group of cysteine is then that you have a methylene group with a thiol group, like so. Now what's going to happen is you're going to bring over the palmitic acid molecule, okay, so here it comes. So here's the carboxylic acid group, and then we have the 14 methylene group, so here's a methylene group, okay, and then I'll put a 14 down there, and then you have a methyl group coming off here. Okay, and what's going to happen is you're going to take the alcohol group off the carboxylic acid group, you're going to take the hydrogen off the thiol group, you'll bind those two together, okay, to create water, and you'll then bind this sulfur to the carbon. Now this is very similar to an ester link. If this sulfur atom had been an oxygen atom there, then we would call this an ester link because we've linked an alcohol group to a carboxylic acid group. Now as I've already told you, sulfur is very similar to alcohol, and the thiol group is very similar. Sulfur is very similar to oxygen, and the thiol group is very similar to the alcohol group. So this link is almost very similar to an ester link, basically, and therefore it's called a thioester link, okay? So it's an ester that involves sulfur rather than oxygen. Okay, right. And that's how you add these palmitoyl groups onto cysteine residues, which are within the middle of the polypeptide. Okay, and this is why it's called S-palmitoylation, because we are adding palmitoyl groups onto the sulfur atom of the cysteine amino acid residue. Okay, right, and palmitoylation is just the process of adding palmitoyl groups onto the protein. Right, okay, so that's S-palmitoylation. There is also N-palmitoylation, which is almost identical to N-myristorylation. Okay, so remember, in order to add a myristic acid molecule onto a protein, you had to have a glycine amino acid at position 1 of the polypeptide, and you could then form an amide link between the carboxylic acid group of the myristic acid molecule and the amino group of the glycine molecule, and you could do that because the amino group was free, because it wasn't bound to the carboxylic acid group of the amino acid before it, because there is no amino acid before it. Now, if you have cysteine amino acids at position 1, then basically you can add palmitoyl groups onto the free amino group of a cysteine amino acid. So let me show this. So here now is the structure of the cysteine amino acid that is at position 1. So it has its amino group intact basically, even though its carboxylic acid group is then linked to the amino acid that's next to it, the next amino acid along. Okay, so what's now going to happen is you're going to bring the palmitic acid molecule over here. So here's the alcohol group of the carboxylic acid group, and then let's put in the 14 methylene groups here. So it's a little squashed, and I apologize for that. Okay, and then there's the methyl group here. And again, what you're going to do is it's exactly the same reaction as for myristic acid. You're going to take the hydrogen and the alcohol group off, and then bind the carbon to the nitrogen to form an amide link there. Okay, and when you have a palmitoyl group dangling off the amino group of the first amino acid in the polypeptide, which happens to be a cysteine, and it needs to be a cysteine for you to be able to add palmitic acid molecules onto it, then that's called N-palmitylation. Okay, right. Now, basically, having palmitoyl groups stuck onto a protein targets that protein for the inner leaflet of the phospholipid bilayer. So let me turn over the page and discuss this. So basically, if we draw the phospholipid bilayer here, so the plasma membrane or the cell membrane consists of 2 layers of phospholipids. Okay, the outer layer of phospholipids, which faces the extracellular fluid, is called the outer leaflet. Okay, so this portion here would be considered the outer leaflet of the phospholipid bilayer, and the inner layer of phospholipids, which faces the cytoplasm of the cell, this would be called the inner leaflet of the phospholipid bilayer. Okay, so this is the inner leaflet. Now, basically, if you take a protein, let's say we have a protein here, which I'll just draw as a square, and then you add a palmitic acid molecule onto it. Okay, so let's say this is a palmitic acid molecule onto it. And I want to stress that unlike myristoylation and also prenylation, you can add multiple palmitoyl groups onto proteins, so we could put more than one basically on. What happens is these palmitoyl groups target the protein for the inner leaflet of the phospholipid bilayer, so the palmitoyl groups will sort of latch into, they'll anchor into the inner leaflet of the phospholipid bilayer because they are very hydrophobic, and so are the phospholipid tails within the phospholipid bilayer, and that will hold the protein at the inner leaflet of the phospholipid bilayer. Okay, so these proteins that end up being palmitoylated are generally very important in transmembrane signaling, basically, in conducting signals across the membrane, so they're generally involved in signaling cascades. Okay, so these proteins are generally involved in transmembrane signaling, which just means across the membrane signaling. So getting a message that's outside of the cell to have an effect that's inside the cell. Okay, and what's very interesting is that palmitylation is reversible. So remember, this is another difference between palmitylation and myristoylation. Once you have added a myristoyl group onto the first glycine of a polypeptide, there is no taking that off. Okay, whereas when you add palmitoyl groups onto proteins, there is an opportunity to remove them. Okay, so there are enzymes which are called palmitoyl thioesterases. Okay, so they are palmitoyl thioesterases which can remove palmitoyl groups from proteins. Okay, and what would happen if a palmitoyl thioesterase came along and ripped these palmitoyl groups off this protein, hydrolyze the thioester links? Well, the protein would now dissociate from the palmitic acid molecules, and it would go back off into the cytoplasm. So what would that mean for the signaling transduction that this protein was taking part in? Well, if the protein is no longer at the inner leaflet of the phospholipid bilayer and is now in the cytoplasm, it may not be able to participate in the signaling cascade anymore. So potentially these palmitoyl thioesterases are extremely important in regulating the amount of these crucial proteins in signaling cascades that are actually functioning, and therefore potentially they could be involved in, for instance, desensitization of receptors, for instance, stopping receptors from being able to induce an intracellular effect. Okay, so that's why there's a lot of interest in palmitylation of proteins. Okay, now, and we'll talk a lot about palmitylation in upcoming videos. But for now, we will turn our attention from palmitylation to the final type of lipid modification that you can make to proteins, okay, which is adding glycosyl phosphatidyl inositol groups on. Okay, so we'll turn our attention to glycosyl phosphatidyl inositol linked proteins. Okay, so to explain what these are, I'm going to start off by explaining what a phosphatidyl inositol molecule is. Then we will extend that up to a glycosyl phosphatidyl inositol molecule, and then finally we'll see how this allows you to attach a protein to the glycosyl phosphatidyl inositol molecule, and then anchor that protein at the plasma membrane. And glycosyl phosphatidyl inositol anchors proteins on the outer leaflet of the phospholipid bilayer. So, let's have our phospholipid bilayer here again, and we're now talking about the outer leaflet. Okay, so let me show you the structure of a phosphatidyl inositol molecule. Okay, let me just move this up as well. So, firstly, we'll start off with the structure of a normal old boring old phospholipid, and then we'll modify it to produce phosphatidyl inositol. Okay, so this cartoon here represents the structure of a normal old boring old phospholipid. So these two vertical lines here, these represent the hydrophobic tails of the phospholipid. Okay, and these are the long-chain carboxylic acids, or the fatty acids, because the proper chemist name for a fatty acid is to call it a long-chain carboxylic acid. Okay, so it just means a carboxylic acid with a very long tail. And myristic acid and palmitic acid are examples of fatty acids. They're very long-chain carboxylic acids. Their tails, remember, consist of either 13 or 15 carbon residues. Okay, so let me colour in those long-chain carboxylic acids in green. Okay, and those are anchoring the phospholipid in the phospholipid bilayer. Then the basis for the entire phospholipid structure is this molecule here, which is represented by this orange horizontal line, which is the molecule glycerol. Okay, and the proper chemist name for glycerol is to call it propane 1,2,3 triol. And although propane 1,2,3 triol is a mouthful, it's useful because it tells you exactly what this molecule is. It is a 3-carbon molecule with alcohol groups coming off the first, second, and third carbon. And off each carbon you have just one alcohol group coming off. Now, you then have long-chain carboxylic acids esterified to the alcohol groups off the first and second carbons. And then off this alcohol group coming off the third carbon, you then have a phosphate group attached there. Okay, so this little purple ball here, this represents a phosphate group. Okay, and this phosphate group is attached to the alcohol group of the glycerol molecule, the third alcohol group of the glycerol molecule specifically, via a phosphoester link. Okay, so that's very similar to an ester link as well, except that instead of using a carboxylic acid, you use a phosphate group. Okay, so instead of being between a carboxylic acid and an alcohol, this time it's between a phosphate group and an alcohol. But phosphate groups, if I just show you the structure of a phosphate group, this is the structure of a phosphate group. You have a phosphorus atom with an oxygen double bound to it. You then have an alcohol group, and you can see that this looks awfully like a carboxylic acid group, except that you've got a phosphorus atom here rather than a carbon. And then you have another alcohol group, and then an oxygen that's linked to it by a single bond, and which has also acquired an electron via ionic means, and therefore has a negative charge. Okay, so basically you can use this just like a carboxylic acid group here and attach this via a phosphoester link to an alcohol group, and that's what you've done here. Now, once you've used this alcohol group to form a phosphoester link, you still have this other alcohol group, so you can form another phosphoester link. And that's what this phosphate group is going to do. So this structure is the structure of a normal old boring old phospholipid. Okay, and I'll just call this a phospholipid, but there is another name for a phospholipid, an old biochemist's name for a phospholipid. Okay, and the other name for a phospholipid is to call it a phosphatidate molecule. Okay, and although no one would ever refer to a phospholipid as a phosphatidate molecule, when we're talking about modified phospholipids, we use phosphatidyl all the time. So phosphatidyl just means a phosphatidate molecule with something else stuck onto it. So we want to talk about phosphatidyl inositol, which is a phosphatidate molecule with inositol linked off it. Okay, so inositol is a 6-membered ring, and I'll put it here. Okay, so it's a 6-membered carbon ring where all of the bonds between the carbons in this ring are single bonds, and then each carbon in this 6-membered ring also has an alcohol group coming off it and also a hydrogen coming off it. Okay, and what's going to happen is this carbon here is going to use its alcohol group to form a phosphoester link with the remaining alcohol group of this phosphate group here. And this structure here now, where we have a phosphatidate molecule linked to an inositol ring, so this is the inositol ring, this is known as a phosphatidyl inositol molecule. Whoops, inositol. Okay, right. Now, we're not going to stop there. That's the structure of phosphatidyl inositol, which is often just abbreviated to PI for short. P for phosphatidyl and I for inositol. So far what we've discussed is phosphatidyl inositol, this bit here. But we want glycosyl phosphatidyl inositol, so we want to add a few more things on here. Okay, so basically, off this carbon here, you are now going to bind 4 monosaccharides. Okay, so let me show these. Now I'm not going to draw their structures out, I'm just going to represent them as boxes to try and keep this as simple as possible. Okay, so here we have 4 monosaccharides bound together to form a little tetrasaccharide here. So this is what's known as a tetrasaccharide, and I'm just wondering where I'm actually going to draw the rest of it, because ideally I would have had more space up here, but I don't, so I'm going to have to draw the rest of it over here now on its side, okay, which isn't ideal, but I just now need to make sure I keep that space free. So, tetrasaccharide, okay, now which monosaccharides does it consist of? Well, I'm going to have to color code them and label them down here rather than labeling them here because I need that space. So we'll start off with this one that I've just colored in orange here. So this is a glucosamine monosaccharide here. So it's glucose, where the alcohol group of the second carbon has been replaced by an amino group rather than an alcohol group. Okay, so that's in orange there, and the other 3 are all the same basically. So I'll put these here, I'll highlight these in pink. Okay, so these 3 then are mannose, okay, which is an optical isomer of glucose basically. Okay, so here is mannose in red. So we link 1 glucosamine to 3 mannose molecules, and then we're going to link this final mannose over here to a phosphate group, okay, which is then linked to an ethanol-amine molecule. Okay, so I'll show this here, like so. Okay, so here is the 2 carbons of the ethanol molecule, and then it also has an amino group over here. So basically, this molecule in the middle here is a phosphate group, and again what has happened is the phosphate group has formed a phosphoester link with this with an alcohol group of this mannose molecule here, and it's also formed a phosphoester link with the alcohol group of this carbon of the ethynylamine molecule. So ethynylamine would have an alcohol group coming off this carbon here. So this is ethynylamine. Okay, and when you have a phosphate group like so, linking two structures together which both have alcohol groups, I should have said this before when I talked about glycerol and inositol, that sort of a structure is known as a phosphodiester link. Okay, so you say that the mannose and the ethynylamine are linked by a phosphodiester link. Okay, and you would say that the inositol and the glycerol are linked by a phosphodiester link. Okay, so you have two phosphoester bonds. This is a phosphoester bond, and this is a phosphoester bond, but the entire thing would be called a phosphodiester. Okay, now, finally then, this is the structure of a glycosyl phosphatidyl inositol molecule. So the entire thing now is GPI, and I hope what you can appreciate is that it is just a modified phospholipid. It's a phospholipid with a massive bit stuck on its head. Now, how is this going to attach to proteins? Well, it's going to be this amine group of the ethynylamine over here that's going to then link to the carboxylic acid group of a protein. So, remember when we were discussing the structure of proteins? Proteins are this polymer of amino acids. Okay, loads and loads of amino acids polymerized together, and you have two terminals. You have the carboxy terminus, which has a free carboxylic acid group, and you have the amino terminus, which has a free amino group. Okay, and basically, when we add glycosyl phosphatidyl inositol molecules to proteins, we're going to bind the free carboxylic acid group here to this amino group of the glycosyl phosphatidyl inositol. So, if we have our free carboxylic acid group here of the protein now, and then the rest of the protein is over here, okay, what we're going to do then is we're going to take the alcohol group off the carboxylic acid group, we're going to take a hydrogen off this amino group, and then we'll bind the nitrogen to the carbon to form a amide link. And that's how you anchor proteins to glycosyl phosphatidyl inositol molecules to create a glycosyl phosphatidyl inositol linked protein. And I would like to somehow show this. So what's going to happen is you're going to take off one of those hydrogens. I'll reduce it down to a single hydrogen off there now, and we'll bind this nitrogen to this carbon, and of course you're going to break that bond there. Okay, so that will create a secondary amide link between the protein and the glycosyl phosphatidyl inositol molecule, and the protein will now be anchored in the outer leaflet of the phospholipid by there and will face out to the extracellular fluid, okay, and it will now be known as a GPI linked protein. So that now concludes our discussion of the basics of lipid modification of proteins.